Hello everyone, welcome to Biology 101. Today we're going to be learning about critical thinking skills that are absolutely necessary to understand biology and also how scientists conduct research. Okay. Well, all research is on the basis of scientific inquiry. Scientific inquiry is essentially scientists asking questions to come up with answers to natural phenomenon. Scientists will ask questions and then use the scientific method to conduct experiments and research in order to figure out answers to almost anything, from why is the grass green to what's a subatomic nuclear particle do. So scientific inquiry is essential to not only advance the discoveries that we're now seeing in science, but also to expand the knowledge of what we once understood in the sciences and what we understand today. So all scientists follow the scientific method, and the first step always, according to the scientific method, is to ask the questions. What questions do we ask? Well, all research starts with trying to answer a question about a natural phenomenon in order to expand our knowledge on what would happen if certain parameters were changed. For example, we all know that plants need water, but how much water exactly does a plant need? Well, that would depend on the kind of plant and the soil and many other factors. Well, what if a researcher wanted to figure out if there was an ideal amount of water needed for each kind of plant? Well, they could ask the question, how much water does, for example, a rose bush need in order to grow optimally? So let's say that becomes the question. So if that's the question, what needs to be addressed next is, okay, if I change the amount of water to, let's say, 30 or 40 milliliters, what would happen to the rose bush? So that is the kind of questions that we're looking at. And it helps us narrow our focus towards, what it is that we're looking to discover. Before any experiment can be conducted, background research must be done, because you never know if there are other experiments previously conducted that answer the question at hand. And also, using the information gathered from previous experiments, a scientist can plan out how they want to conduct their experiment, and if there's anything that was tested in previous experiments that were proven false later on. So using a literature review or going through multiple research projects that talk about the parameters of what you're trying to research is extremely important to not only designing the experiment, but making sure that what you're researching can actually be proven via conducting experiments. The first step after this is to begin the process of designing the research project. What we have to do first is create a hypothesis. A hypothesis is essentially a theory that we use or that we create in order to explain why we feel a certain phenomenon is occurring. An example of a hypothesis could be, if I add 40 milliliters of water to a plant every single day, then it should grow optimally. Now, this is something that can be tested, and the variables in the experiment, which I'll talk about next, can be manipulated in order to figure out, okay, if I add 40 milliliters of water, I feel that's ideal for the plant. But what happens if I do 30 milliliters versus if I do 50 milliliters? Typically, one experiment, though, is used for each hypothesis. This doesn't mean one trial. An experiment can have multiple trials and multiple variables. However, if I'm going to conduct an experiment that talks about the amount of water that's necessary for a plant, then I'm only going to create a hypothesis for that experiment that talks about what I think will happen to the plant and what I think the best amount of water is needed, best amount of water needed for a plant. So now we can conduct the experiment after putting out our if-then statement. If I add 40 milliliters of water, then the plant should grow optimally. Okay. During the experiment, we also need to find a way to test the hypothesis that we're creating. And so experiments are conducted in order to test different hypotheses. So in an experiment, we have variables. Variables are studied during the experiment and are essential to figuring out the answer to our question. There are two types of variables present: independent and dependent variables. An independent variable is an experiment condition, like, for example, for a plant, I would say, okay, one of the independent variables could be the amount of water that is being put in order to hydrate the plant. So this is something that we will have already planned out before we even conduct the experiment. So we'll have one plant that is taking in 40 milliliters of water, and let's say another plant that's doing 30 milliliters and another one that's doing 50 milliliters. This is a variable that was already determined before we even started running the experiment. The dependent variable would be what results from the changes that we made with the independent variable. So what's going to happen to the plant at 50 milliliters of water, 30 milliliters of water, and 40 milliliters of water is the dependent variable. So essentially the effect of the changes that we made to the independent variable. So after we determine our variables and what we're trying to look for and how we're going to determine whether our hypothesis is correct, we have to collect data. So there are four main types of research data that can be collected. Observational, which involves data collection by observing or looking at a behavior or activity and writing down exactly what we're seeing happening in a natural setting. Then there are experimental data, where data is collected through conducting an experiment, and this involves direct manipulation by the scientist, meaning that the scientist itself has to try to recreate this natural phenomenon rather than just observing it in nature. And oftentimes this involves manipulating different variables, such as, for example, with a plant, there could be controls and variables. So you would have, sunlight or soil be a main variable that could be manipulated in addition to water. So here we would be using an experiment to figure out, are there other factors that help explain why 40 milliliters of water could be the most important for plant growth? So here we would collect data based on what we were experimenting with. Simulation is data that's collected by computer simulations of a real-life situation. So let's say we don't have the resources to figure out how much water a plant needs. Well, what if we could take the data in a computer setting and simulate it with different plants mathematically to figure out what is needed in order for plant growth? Funny enough, actually, a lot of simulation labs can do this, where you can pull up multiple plants and then see how much is necessary in terms of water, soil, sunlight to keep it growing. That way you can test out different parameters throughout the experiment, but keep the focus on, how much water is necessary. The last type of data that could be collected is derived or compiled data. This is data collected by using existing data points and aggregating it under something called an algorithm. What does this mean? So let's say we have data points that were conducted from someone else's previous experiment. We would take that data and let's say we're only looking for the results that came from plants that had 40 milliliters of water. So we can pull this data from the previous experiments from other researchers or previous experiments that the scientists conducted and kind of put it all under one aggregated area where we can now compare just what's happening to the plants that got 40 milliliters of water. So this is definitely a way of conducting an experiment where we're just trying to narrow down the focus towards the kind of effects that we're looking for without having to physically conduct the experiment ourselves. After collecting data, the next step that we would have to take is analysis. Analysis of a result or multiple results is absolutely necessary for understanding what exactly we just observed. The thing is, the best analysis involves comparing what has happened from what experiments that we've conducted and data we've collected and comparing it to what we predicted earlier in the hypothesis. So let's say in the hypothesis from before, I said, if 40 milliliters of water is used, then the plant will be at optimal health. Okay, well, then I would look into my data. And what I would see is, okay, when I used 40 milliliters of water, was the plant at optimal health? If so, then my conclusion matched my hypothesis. If not, then that means that the hypothesis does not support what I originally thought was the ideal amount of water necessary to help the plant grow. And now I need to go back and redo my experiments or change some variables or propose a new theory that can allow me to really figure out the answer. So analysis is extremely important. Let's say that the conclusion does support the hypothesis. This is now information that can be used by the scientific community. But even if it does support the hypothesis, it's always important to conduct multiple studies, multiple experiments, to see if the results can be replicated by other scientists. If the results can be replicated, then this means that this was solid research that now can be published in something called a research journal. So after results of experiments are published, these results will be evaluated by the rest of the scientific community via journals, and this helps to expand the knowledge that is originally thought of for a different phenomenon. And so many of times, results from research experiments are very helpful to many different fields. For example, clinical studies and their results, where certain medications are tested out or certain therapies are tested out, are essential to helping physicians make informed decisions on a patient's health or behavior. What do I mean by this? Well, let's say there's a new drug that's being put out in the market. There needs to be multiple trials conducted before that drug can actually be given to another person. And so the clinical studies of this drug trial would be to test it first on animals and then see if they're okay and then test it on humans. And then only after multiple testing and revision and checks for safety can that drug be marketed to everyone else. And sometimes there are miracle drugs like penicillin, for example, which was created to cure antibiotic-resistant bacteria, was absolutely created in order to prevent illnesses. And this actually changed a lot of, the livelihood of a lot of people who would die from illnesses that can now be cured by penicillin. And so what I feel is, even if there's a disproved hypothesis, it's always good to share the results, because this allows more research into why the results were false. And so often studies and publishing in journals don't typically put out disproved hypotheses in their journals. However, scientists should put out, in my opinion, disproved hypotheses because it's essential to the expansion and growth of the knowledge in the scientific community. Now, that is actually the last step of the scientific method, all the way from going to creating and asking a question and then publishing the results. And so, results of experiments and also the whole experiment itself laid out in journals is what helps advance the scientific community and hopefully helps us advance our knowledge on biology and how the world works. Okay, so now I'm going to head into the knowledge check. Hopefully you guys can take a few minutes to figure out your answers, and I'll give you a minute or two, but if you need more time, feel free to pause the video, and then we will move on to the answers to the knowledge check. Okay, these are the answers to the knowledge check. I'll give a few minutes so you can check over your work, and we'll move on. And this is the end of our presentation on the scientific method and the critical thinking skills that are needed in order to comprehend and conduct scientific research. Thank you.